Very good afternoon one and all. This is your Prasanna Ma'am from NEET World. So yes, in our previous class we have clearly dealt with the syllabus breakdown of biology subject in the exam NSEJS. Okay. So in this exam we have discussed completely about the syllabus breakdown under biology, and from today we are going to start the questionnaire session of NSEJS paper, wherein we are going to focus on each and every concept that is given over here. Okay. How we are solving the question paper here is, first let us look into the question in detail, and then we will analyze the concept behind each question. As such, we will cover multiple concepts related to each and every question. Clear, right ma? So yes, let us begin with the first question. In the flow chart given below, accumulation of various products from 1 to 4 results in some inborn diseases in man. So first, whichever question we are solving, let us think from which concept of the syllabus the question is asked. So as it is highlighting the inborn diseases, this question is clearly from which chapter beta? This question is from heredity. Okay, wherein in this question there are multiple metabolic pathways that are given. Okay. So looking into this multiple pathways, you need not get tensed or confused, children. First, you break it down in this way. For example, so 4 metabolic pathways are highlighted in this sequence of the metabolic pathways. 4 processes are highlighted. This is 1, 2, 3 and 4. 4 processes are highlighted. So at these steps, which is getting accumulated because of what enzyme deficiency leading to which inborn disease, that should be identified. So here if you have to look into beta, first this is what metabolic pathway? This is phenylalanine metabolism, wherein phenylalanine has to get converted to tyrosine with the enzyme phenylalanine hydroxylase. Okay, phenylalanine hydroxylase. Okay, so this enzyme will catalyze the reaction from phenylalanine to tyrosine. If this phenylalanine hydroxylase is deficient, then phenylalanine will get accumulated as phenylpyruvic acid, which results in the disease phenylketonuria, which is an inborn disease. Okay, this will result in phenylketonuria. Okay, and the next is tyrosine metabolism, wherein tyrosine gets converted to hydroxyphenylpyruvic acid. So the disease tyrosinases. Okay, the second disease will be tyrosinosis. Okay, second one is tyrosinosis. Just a second. Yeah. So the second disease is tyrosinosis, wherein this disease occurs because of the accumulation of one product in tyrosine metabolism. Okay. So in tyrosine metabolism, tyrosine getting converted to p-hydroxyphenylpyruvic acid, this gets accumulated. Fine. And here which enzyme is deficient? P-hydroxyphenylpyruvic dioxygenase. Okay, this enzyme is deficient. So this causes the disease tyrosinosis. Okay, second one, this accumulation will cause the disease tyrosinosis. Fine, and this is because of the deficiency of what enzyme? P-hydroxyphenylpyruvic acid dioxygenase. Clear? Next, at this step, homogenetic acid is accumulated. This is because of the deficiency in the enzyme homogentisic oxygenase enzyme, oxidase enzyme. homogenetic oxidase enzyme, because of the deficiency of that homogenetic acid gets accumulated and this results in the inborn disease alkaptonuria. Remember that. This gives what disease? Alkaptonuria. Fine. And then DOPA to melanin, conversion of this. Okay. DOPA, if it gets accumulated, melanin formation will not happen beta. So, DOPA, if it is getting accumulated, melanin formation does not happen. Because of the accumulation of DOPA, what is it resulting in? Albinism. Clear, right? Once again, let us quickly recall the question. First is phenylalanine metabolism, wherein phenylalanine has to get converted to tyrosine, but then this is getting stored in the form of phenyl pyruvic acid. Okay, it gets stored in the form of phenyl pyruvic acid because of the deficiency of what enzyme? Phenylalanine hydroxylase. Phenylalanine hydroxylase. Fine. And second is accumulation of hydroxyphenyl pyruvic acid from tyrosine. This is because of deficiency of what enzyme? P-hydroxyphenyl pyruvic acid dioxygenase. Next is because of the accumulation of homogenetic acid because of the deficiency of the enzyme homogenetic oxidase. This results in the disease alkaptonuria. And then because of the accumulation of DOPA, melanin formation does not take place and this leads to the disease albinism. Once let us clearly look into the explanation also, ma. Yeah. So, here first disease is phenylketonuria, where phenylalanine is accumulated in the table as I told you. This gets accumulated due to deficiency of what? Phenylalanine hydroxylase. This phenylalanine hydroxylase will convert phenylalanine to tyrosine and this results in the disease of what? Phenylketonuria. Second is tyrocyanosis. This is because of the accumulation of P-hydroxyphenyl pyruvic acid in tyrosine metabolism. This accumulation leads in the disease of tyrocyanosis, which is a metabolic disorder due to tyrosine degradation. Next disease is alkaptonuria. This is because of the accumulation of homogenetic acid. This is a genetic disorder where homogenetic acid oxidase is deficient. And after this the disease is albinism, which is a precursor, DOPA gets accumulated, which is a precursor of melanin. Melanin formation does not take place. If tyrosinase enzyme is deficient, then melanin is not formed. This results in the albinism. Clear, right? So, this table we have to remember, ma. In this, what will be the right answer? First disease is phenylketonuria, second is tyrocyanosis, third is alkaptonuria and fourth will be albinism. So, option A will be the right answer. So, in this, yeah, this table is important, where from this question we can take the references. We can take the references of this enzyme deficiency. Based on that metabolic pathways, few more questions we can expect in the coming examination. So, because of the deficiency of which enzyme phenylketonuria occurs? Because of the deficiency of phenylalanine hydroxylase. Tyrocynemia, which is type 3 disorder, is because of what deficiency in enzyme? P-hydroxyphenyl pyruvate dioxygenase. Third disorder is alkaptonuria. This is because of the deficiency in homogenetic oxidase. Albinism because of the deficiency of tyrosinase. Okay. So, this is all about first question, where we have exercised multiple metabolic or biosynthetic pathways. Okay. Phenylalanine, tyrosinase and DOPA, melanin formation, all this metabolic pathways we have exercised, and because of what deficiencies which disorder is occurring. This you have to highlight beta. Any time in this NSEJS examination, each question of course it is very much lengthy, okay, but then you have to divide it into small chunks so that you can easily decode the question. Understood, ma? Next, yeah. Look at this match the following type of question, where in column 1 all the types of plants are given, and in column 2 the characteristics are provided. First, if you see wheat, okay, which is a monocot. In monocots, and wheat belong to which family? Poaceae family. Wheat belongs to Poaceae family. Okay. So, here in this family they will be having glumes and lodicules. Okay. What is glumes? They have what type of inflorescence in Poaceae member? Poaceae members, spikelet inflorescence is present, right ma? Poaceae members will have spikelet inflorescence. So, at the base of this spikelet inflorescence, small leaf bract-like structures are called as glumes. Glumes are nothing but small leaf-like bract-like structures at the base of spikelet are called as glumes. Okay. Lodicules are nothing but, which are the reduced form of perianth. Lodicules are nothing but reduced forms of perianth. Fine. Lodicules are reduced forms of perianth. Next is banyan tree. Okay. Banyan tree will be having prop roots and hypanthodia. Okay. Banyan tree has prop roots, which are adventitious types of roots which arise positively geotrophic from the lateral branches, which gives mechanical support to the plant. Mechanical support. And what is hypanthodia? Hypanthodia is special type of inflorescence. So, hypanthodia is special type of inflorescence. Okay, hypanthodia is special type of inflorescence where it has urn shaped receptacle. Where it has what type of receptacle? Urn shaped receptacle. Just a second. Fine. And then if you see banana, this characteristic feature over here is pseudostem and monocarpellary. Okay. Pseudostem, yes, it has false stem wherein all the leaf sheaths at the leaf base will be accumulated surrounding to the stem region. And here this is called as pseudostem because the stem is not made up of woody tissues, they are surrounded by the base of leafy sheaths. That is why it is said to be pseudostem, and it is monocarpellary. Okay, it is monocarpellary. And last is pine tree. Pine is what? A gymnosperm, right ma? So, pine tree, which is a gymnosperm. So, this pine tree is having what? Seeds but no fruits, because in gymnosperms ovules are present but ovary wall is absent. Ovules post fertilization will turn into seed, ovary wall to fruit. As ovary wall is absent in gymnosperms, fruit formation does not happen, and in gymnosperms you see what ma? Naked seeds. Clear, right beta? So, here wheat, which are Poaceae members, they show glumes and lodicules. Glumes are nothing but the bract-like structures which are at the base of the spikelet inflorescence. Lodicules are nothing but the reduced form of perianth which you see in the Liliaceae members. So, the reduced form of perianth is called as lodicules. These are glumes and lodicules are the characteristic features of Poaceae members. Remember that. Next, banyan tree. The characteristic feature of banyan tree includes prop roots and hypanthodia. Prop roots will give mechanical support. Prop roots are also called as pillar roots. They arise from the lateral branches towards the ground, which grow positively geotrophic, and they aid in mechanical support to the main plant. Hypanthodia, which is a special type of inflorescence, it has urn shaped receptacle. These two are the characteristic features of banyan tree. Next is banana, it has pseudostem because at the base, because the stem region is not formed by woody tissues, it is formed at the base of leafy sheaths, and then reduced and then it is also having monocarpellary feature. Pine tree, which is a gymnosperm, it has seeds but no fruit. Okay, pine tree, which is a gymnosperm, it has seeds but no fruit, wherein ovule turns to seed, which is snake. Ovary wall is not present in the gymnosperms. Clear, right? So here what option we have to choose, ma? One is 2, then banyan tree, 1, 2, 3, 4. Banyan tree, which is 2, is option 4, then 3 is 1 and 4 is 3. Option C is the right answer. Next, so here I have tabulated the complete explanation beta. So in this table it is clearly given, see, wheat has characteristics for glumes and lodicules, where wheat is a grass, belongs to Poaceae family, monocots. Its flowers have glumes and lodicules. Glumes at the base of the inflorescence and lodicules which are the reduced form of perianth. Banyan tree, if you see, it has prop roots and hypanthodia, wherein banyan, that is ficus, develops prop roots or pillar roots for the support or mechanical support or aid, and it has hypanthodium, which is a special type of inflorescence with urn receptacle. Next, banana. Banana has characteristic is what? Pseudostem and monocarpellary. Banana has pseudostem and flowers once in the entire lifetime, that is monocarpic. Okay, banana flowers only once in the entire lifetime, so it is monocarpic. Pine tree, if you see, seeds are there but no fruits. Pine, which is a gymnosperm, this is producing seeds without any fruit, so it is nectar. And then next question is from molecular biology, which is on the nucleosome. So in eukaryotes, what happens? DNA packaging takes place. The packaging of the DNA is explained in many models. One of the best model is nucleosome model. So here in the question, it is about, under the electron microscope, chromatin appears as a string of bead like structures. Each repeating unit is called as nucleosome. Consists of 5 components, those are histones. Parts labeled 1 and 2 in the diagram will represent the histone proteins forming the core and the linker. So here if you see, ma, the DNA which is negatively charged, it will be the chromatin thread like structure. Okay, DNA which is chromatin thread like structure is condensed with the help of positively charged histone proteins. So histone proteins here are, histone proteins are positively charged. These are positively charged because of the basic amino acids, those are lysine and arginine. Lysine and arginine. Fine. So histones which are positively charged proteins, they have basic amino acids, they are composed of basic amino acids, lysine and arginine. And DNA, of course we know DNA is negatively charged, because of phosphoric acid, that is H3PO4. Now this negatively charged DNA is condensed or packed by positively charged histone proteins like beads on string. So each time the DNA is wrapping around this, so here this is the DNA chromatin thread, it is wrapping around the histone octamer. So this complete chromatin structure is observed or visualized like beads on string model. Okay, this is visualized as what, ma? Beads on string model. Understood, right? So here you see, choose the correct option showing the correct sequence of 5 components. Fine. So here this histone octamer which is at the center, okay, just a second. Yeah, this histone octamer which is at the center where DNA is being wrapped. Histone octamer is having, histone octamer will have 2 sets of 4 histone proteins. Those are H2A, H2B, H3 and H4. Okay, 2 sets of this are present, H2A, H2B, H3 and H4. So, in this histone octamer which are total 8 molecules, 2 sets of 4 histone proteins. They are present. Around this the DNA is wrapped and it leaves out. Okay, this DNA which is wrapped will leave out and this connects to another nucleosome. This is called as one nucleosome, and the linker DNA which is connecting 2 nucleosomes is called as linker DNA, and here H1 histone proteins will be present. Linker DNA which has H1 histone proteins. So, in the histone octamer how many are present ma? 4 histone proteins. How many sets? 2 sets, and here what are they? H2A, H2B, H3, H4 are present, and the DNA which is coming out of this nucleosome is linked with one more histone protein. What is that? H1 histone protein, and that DNA is called as linker DNA which connects the 2 nucleosomes. So, now you see, choose the correct option showing the correct sequence of 5 components. So, in 1 and in 2 we have to identify. First one, what is there? H2A, H2B, H3, H4, and in this linker DNA what protein is there? H1 histone protein is present. So, option C will be the right answer over here. Clear, right ma? Next. Yeah. So, this is the explanation that I have given till now, that in this complete model, if you can see, in this complete model the DNA is wrapped, DNA which is negatively charged is wrapped around this positively charged histone proteins which are total 8 molecules, and it has how many sets beta? 2 sets of histone proteins, H2A, H2B, H3 and H4, and this linker DNA is having H1. So, nucleosome bead will have 8 histone molecules and 146 nucleotide pairs of DNA. Till here it is clear, right ma? Next. So, the last question for today's session is taken from the chapter Sexual Reproduction of Flowering Plants. So, this is from the chapter Sexual Reproduction of Flowering Plants. Sexual reproduction of flowering plants. If you clearly see this, in the diagram given below, what are the maximum kinds of ploidy of various parts are labeled. So, before looking into the ploidy of the question which is given, just listen to the explanation of male gametophyte of the angiosperms. Let us get started. So, here if you look into this is the male reproductive structure in angiosperms. Okay. Male reproductive structure. What is the male reproductive structure? Stamens or androecium. Androecium or stamens. So, in most of the angiosperm, the stamens are having bilobed anther. First, look into the stamen structure. It has the filament, the long filament and bilobed anther. Can you see? It has a long filament and a bilobed anther. This long filament and bilobed anther, you see, filament, this is the distal end and this will be the proximal end. Remember, this is what? Proximal end, and this is the distal end and this will be the proximal end. Fine ma. The proximal end of the filament, this is long filament, right? Filament or stalk of the anther. So, this long filament proximal end is attached to the receptacle and the distal end is attached to the bilobed anther. Remember how many lobes it has? Bilobed. Most of the angiosperm will have bilobed anther only. So, the male reproductive structure which is andrecium or stamen will be having what ma? A filament and a bilobed anther. This filament or stalk, the proximal end is attached to the thalamus and the distal end is attached to the bilobed anther. So, this bilobed anther will have how many lobes? Of course, the name itself is bilobed. So, it is having 2 lobes, lobe 1 and lobe 2, and it has 4 theca. Each lobe is having 2 theca, theca 1 and theca 2. Here also it has 2 theca, theca 1 and theca 2. Okay. So, bilobed anther and tetrathecus. Remember the terms, very important, Nana. Bilobed and tetrathecus. Fine, bilobed and tetrathecus. And each anther is surrounded by 4 anther wall layers. How many anther wall layers are there here? 4 anther wall layers. First, outermost layer is epidermis. Next to the epidermis it is endothecium. Both epidermis and endothecium are single layered. After endothecium you will be having 2 to 3 layers of middle layers. Okay, 2 to 3 middle layers, and next to that last layer is tapetum. Okay. So, what are the 4 anther layers you have? Epidermis, endothecium, middle layers and tapetum. These are the 4 anther wall layers. So, here if you have to see, epidermis is single layered, endothecium is also single layered. Endothecium will have alpha cellulosic layers, and both epidermis and endothecium will help in the release of the pollen grain, in the pollen grain dehiscence. Next to that you have 2 to 3 layers of middle layers, and next to it you have tapetum. The ploidy, if we have to look into, the sporophytic structures, of course they are diploid, right? Sporophytic structures are diploid in a plant. In an angiosperm, sporophytic structure is diploid, and we know angiosperm is having what main plant body? The main plant body in the angiosperm is sporophyte. So, sporophyte is the main plant body. Sporophyte is diploid. So, when sporophyte is diploid, anther and filament both are diploid. Anther is diploid, so wall layers are diploid. Epidermis is diploid, endothecium is diploid, middle layers are diploid, tapetum is only polyploid in condition. Okay. Tapetum is what? Polyploid in condition. It is because tapetum is nutritive in function. Okay. It provides nutrients to the developing microspores which are present at the center. Okay. So, at the center these are what ma? Sporogenous tissues. At the center these are sporogenous tissues. Sporogenous tissue is also diploid. Sporogenous tissue is also diploid. Okay. So, this diploid sporogenous tissue will be having microspore mother cell. Okay. They will be having what? Microspore mother cell. Microspore mother cells are diploid. Fine. This microspore mother cell will undergo meiosis and they form haploid microspores. Okay. They form what? Haploid microspores. So, here if you see, once again let me repeat quickly, the male reproductive structure is called as andrecium or stamen in the angiosperms, wherein the stamen will have a long stalk called as filament whose proximal end is attached to the thalamus or receptacle and the distal end is having bilobed anther. Okay. Anther has 4 wall layers, wherein the outermost wall layer is epidermis, next to that is endothecium, next to that is middle layers. Middle layers are 2 to 3 layered, and then you have polyploid tapetum. Okay. This polyploid tapetum is nutritive in function, which provides nutrition to the sporogenous tissue which is at the center. Sporogenous tissue will have microspore mother cells which undergo meiotic division and form haploid microspores. Haploid microspores, taking the nutrition from the tapetum, they mature to pollen grains. They will take the nutrients from the tapetum and they mature to pollen grains. Fine? Clear, right ma, till here? So, now you will be in a position to answer this question easily. Okay? So, these are what? Anther wall layers. Now, you answer easily. Endothecium, what ploid it has? Haploid, again, sorry, diploid. All the anther wall layers are diploid. Epidermis, endothecium, middle layers. Tapetum is polyploid, which is the innermost anther layer. So, here endothecium is what? Diploid. Tapetum is polyploid. Middle layers are diploid. Pollen grain is haploid. Pollen grain is said to be the male gametophyte in the angiosperm. Why pollen grains are haploid? Because pollen grains are from haploid microspores. Okay? Haploid microspores will mature and form haploid pollen grains. Okay? Pollen grains are the male gametophytic structures in the angiosperms. Male gametophyte in angiosperms. Remember. Clear, right ma? So, here you see in the diagram given below what are the maximum kinds of ploidies. So, you can see diploid, you can see diploid endothecium and middle layers, haploid pollen grains and polyploid tapetum. So, what will be the right answer, Nana? Haploid, diploid and polyploid. Option D will be the right answer. Clear, right? Yeah. Once you look into this table as well, it will be more clear for you. Yeah. So, here endothecium, which is the layer in the anther wall which is diploid, middle layers diploid which degenerate later part of the anther wall. Next, tapetum, which is nutritive in developing the pollen grains, which is polyploid. It is either tetraploid or octaploid. Next, pollen grains, which are immature male gametophytes, which are haploid. Okay. So, with this we have exercised 4 questions of our NSEJS 2024 paper. Let us continue this each day without fail. If you are attending this session, this will completely finish your preparation of NSEJS, okay, wherein we are covering the questions by highlighting each and every concept. So, that is all for today, children. We will meet tomorrow with again a new set of questionnaire from 2024 NSEJS exam. Thank you.